Let's begin. So hi, welcome to lecture 4. So just a reminder, today is the due date for the first homework. We already received some of the submissions. If not, just get it to us by the end of today. So today we are going to discuss genetic control. And what do we mean by that? Controlling means turning off or on or turning down or up the expression of a certain gene. So it means turning up or down the production of a protein most of the time. And you can do that at multiple steps during the expression cycle, so transcription and then translation, and even after. For bacteria, where control happens is very clearly at transcription. So what it means is the image that you should have in mind is that we have our DNA here. On this DNA, we have some gene, so coding for a certain protein, and here are like some accessories to the gene on the DNA, some accessory sequences. Here's the promoter, and here's the terminator. And what happens when you talk about transcriptional control means that even the transcription of DNA into RNA is inhibited or like regulated up. And we already seen two different types of control that we can have. Was discussed in the first lecture. So in normal times, the gene would be like, this is the DNA, the gene would just be there, the promoter would be free, and so there would just be the cellular machinery that will come, transcribe, and then translate this gene. So it would just be produced with some rate in normal times. And then you can have two different control mechanisms that you can have. One, which is called a repressor, which is some molecule that binds physically onto or near the promoter, and which will inhibit the initiation of the transcription of the gene. So the machinery will come there, and it will not be able to physically get to the start of the gene, just because it's blocked. So in essence, if you have such a repressor, and there are many of them known, if it's present, you turn off your gene. Another possibility is an activator. Which is another molecule that can bind there, and which will actually turn up, which will regulate up the production rate of this gene. So in this case, for transcriptional control, turn up the rate at which the gene is transcribed into RNA. And both happen. So even if we're not talking about bioengineering, just in the cell, these two things do happen. Life has evolved in such a way that both activators and repressors do exist for certain promoters, of course. Okay. And like one of the fun observations about these repressors and activators, so they both have some role to play. Like activator is turning up, repressor is turning down. And you can look into when cells, bacteria, use which kind of control mechanism. And what is a bit counterintuitive, but we will think about this in a second, is that in bacteria you observe activators being used whenever we have a high demand gene. And repressors used for low demand genes. So what that means is a high demand gene is something that's turned on most of the time. And a low demand gene is something that is turned off most of the time. Which is a bit counterintuitive because you could think in terms of energy preservation that for a low demand gene, most of the time maybe it's very lowly expressed. And only if you want to turn it up, then we have to spend the energy to get the activators to the promoter and turn it off. But it's exactly the opposite that we see. And this is kind of a small graph about this. Like here we have a repressor, and low demand means over time there's only like a small percentage of the time or smaller percentage of the time where it's actually turned on, where there's no repression going on. And activation is exactly the opposite. So most of the time we have to spend the cell does spend the energy to get the activators there and activate it. It's not completely solved why that is. It seems counterintuitive. One of the possible explanations is this use it or lose it kind of way of thinking, is that let's say you have a low demand gene, so it's turned on just a small fraction of the time. You would have a higher propensity of deleting your repressor out of your genome, of mutating it to a lower efficient variant, because you just don't use it most of the time. Most of the time you don't need the gene anyway, so the repressor might be mutated away or just turned down. So, which is one possible explanation of why a low demand gene does have repression going on. And counterintuitively, once more, because you have to spend the energy all the time. Okay, then let's look into a bit of mathematical modeling of control. You can put it there. And we start out with a model that we've already seen in the first lecture, which is just this constant production rate of a gene. So let's say X is our protein concentration, and we just have some beta, and with some degradation rate gamma. Okay. There's no control going on at the moment, it's just production. But one question that you can ask is like, can you control the speed of gene production? Of protein production in this sense. And the first thing that you have to do if you want to ask the question or to try to answer the question is to measure the speed here. So let's first try to see what happens here. So of course for this simple system, I think we already analyzed it, but we can do it again here. The first question to ask often if you encounter like an ODE model is to ask about the steady state. Where does it converge to? And here, of course, we get beta over gamma as the steady state concentration. Okay. Here, in this specific case, we can even solve the ordinary differential equation explicitly. Because this is an affine equation. So we know that this will be actually beta over gamma 1 minus e power minus gamma t. Okay. So it approaches exponentially fast the steady state. at time t, of course, at t equal to 0. This will be 1, so we start out at 0 for this solution. Okay, so what will happen? Just a small picture. Time. This is x. There will be this limit, beta over gamma. And we will converge somehow in this sense. Of course, never reaching beta over gamma. And now we can assess the speed of this. Assessing the speed is, again, there are multiple different definitions that are possible. But in an exponential, approach, one interesting time variant is just what is the time until we get beta over 2 gamma? When do we get to the halfway point? And we might call this, I don't know, the rise time, t rise. Okay? And of course, for our example here, we can just figure this out. There's some algebra involved, but it's not too hard, of course. And you can figure out that this is just the logarithm of 2 divided by gamma. Does this make sense? We don't have any beta inside of it. We're independent of the production rate beta. That does make sense because beta here is in a steady state, and we are measuring the time until we get half of the steady state. So it kind of cancels out, and we get only something that is a function of gamma. Okay. If we turn up gamma, the rise time gets smaller. And this is maybe also at first counterintuitive, but this seems to imply that a larger gamma leads to small rise time. Gamma is the degradation rate of our protein. So we're destroying things, but we're making things faster. Does this make any sense? Let's take a look. Here we have a small script, and again, it's in. You can of course play with them if you put these scripts up in Jupyter Notebook. So this would be one expression curve starting at 0 of our gene here. Beta is 100, gamma is 1, so the steady state will be 100 over 1, so it will be 100. The rise time is the time until we reach 50. And now let's change gamma. Let's turn it up. So here we have this multiple rise times. So we have here gamma being either 1, 2, or 3. So we're increasing gamma. So we have 3 curves here. So beta 100 gamma equal to 1 is the original case that we had here. And then we have a bigger gamma and an even bigger gamma here. And what we see, of course, is the steady state also goes down with increasing gamma. But of course, if we normalize again, so this is everything normalized to 1, to the steady state 1. Now we see that the slowest decay is the slowest rise time. And the fastest decay has the highest rise time. So there's a trade-off going on. You might be faster, relatively speaking, but of course your expression is not as high. So turning this up might only make sense if your expression rate was large enough to begin with, so that even turning it up, you have enough of dynamic range of difference between like 0 expression and 1 expression, like full expression. Okay? But relatively speaking, and this is kind of a neat observation, the speed only depends on gamma. Turn gamma up, you have more speed. Now the question is, can we use this information to get some engineering principles about how to use repression or activation if we're only interested in time, like in response time? And for this, you can again look into what bacteria do. You can look into how bacteria regulate the genes. Let's say here, I take an example of E. coli, of course our model bacterium, and give you some numbers. So you see around 424 operons. Operons are things that can be regulated, up or down. You see around 519 regulations. That means genes that can regulate indirectly or directly other genes. And what you see are, and this is kind of also an important observation, is that you get around 40, so around 10% of all the regulations that are going on are autoregulations. That means genes controlling themselves, either up or down. Right? So you can totally have a gene that produces something that represses itself afterwards. Like the gene product, the protein represses itself. And if you zoom into these, you get 32 negative and 8 positive. What does this tell us? So first of all, if you're doing autoregulation, evolution has made it such that negative autoregulation is much more prevalent than positive autoregulation. Okay, we can see why in a second. But also, like, we can also zoom into the 40 here. In some sense, this is a surprising high number of autoregulations. Because you could think of, like, you have 519 regulations going on, and you have 400 like possible targets. So you might think that kind of hitting yourself might be something like 500. The frequency of hitting yourself of autoregulations would be 519 over 424. So 1 point something. But this is not what we see. We see much more than that. So it's not just like a random choice, uniformly random choice, who you would regulate. There's a clear bias towards autoregulation, just in a statistical sense. But apparently does make some sense. So let's look at the most prevalent. First, we look at both today. We look at negative autoregulation and positive autoregulation. Sorry, yes? It's more negative than positive in general, yes. Yeah. Okay. Yes, again, so we will look at both negative autoregulation, positive autoregulation. First, for negative autoregulation. let's first build a model. Again, we had this model in the first lecture. So we can have like X being the concentration of our protein of interest, of our gene product of interest. And let's say, again, we have some degradation rate. And now we have like our production rate beta, but we negatively autoregulate this. So the more X that we have, the less production rate we will get. This is, oh, thank you, yes. X over KD, thank you. Yes. So now if we have, like, imagine X being infinity, you would get 0 production rate, okay? And we derived this, like this is a Hill function, we derived this as the Michaelis-Menten approximation of enzyme kinetics. So this is kind of something that's kind of established as a basis for regulation first. And because we're putting X here instead of something else, this is autoregulation. And because this goes to 0 as X goes to infinity, it's negative autoregulation. OK. Let's try to understand what is going on here. So again, we have an ODE model. ODE model, first question that you can ask is, what's the steady state? The steady state for this is, of course, you set the derivative to 0, and then because you will get the gamma x on the other side, you will multiply by the denominator here, so you will get a quadratic equation, which you can solve. And you get something like we'll divide by 2. And I'm already doing some, or let's not do a factorization right away. Let's do this slowly. So this will be square root of kd squared plus 4 kd beta over gamma minus kd. Okay. The minus solution is the correct one here. Sorry, the plus solution of the root is the correct one here. And now we can ask the question, like, how big is this? Like, we already know that kind of the base steady state would be beta over gamma. That's if there's no autoregulation going on. If this wasn't here, we just divide it by 1. It's a bit different. This will be lower than that because we are regulating ourselves down over time. And it turns out that kind of one interesting regime that we have to analyze this is the regime where this autoregulation is strong. So where this steady state is much lower than just the basal steady state beta over gamma. And this is interesting because it will turn out again that having like strong autoregulation, negative autoregulation, will speed up your system. So you can kind of just by adding autoregulation on your gene, speed up the relative expression rate of your system. Okay. So let's make the hypothesis that beta over gamma is much higher than kd. The picture that you can have in mind here is something like this. This is not, of course, the solution of the ordinary differential equation. But what will happen is, like, if you look at this ODE, at the beginning, when x is very small, like this will not matter. So it's just beta. You're trucking along with beta over time. Then at some point, x is large enough that kind of the denominator will be big, such that your production rate will go down by a big step. This is not true. You don't, like, as soon as you hit KT, as soon as your denominator is 2, you don't completely stop production, but it's a good image to have. And so this would mean that kind of the basal rate, the basal concentration, steady state concentration, beta over gamma would be much higher than what you would expect from this picture, really regulating down. If you're doing the calculations correctly, and you can look up the lecture notes how to do that, it's not very hard. You just take this equation and you're applying this approximation. You will see that the steady state in this regime will be more or less kT, square root of kT beta over gamma, which is much smaller than beta over gamma because the kT is much smaller than beta over gamma. So if you replace this kT by beta over gamma, you would have beta square over gamma square, and you take the square root, which would give you back beta over gamma. So this is much smaller. This is why this makes sense to talk about this as the high autoregulation, high negative autoregulation regime. Okay. Now this is the steady state. You can also look at the dynamics here. So really at the approximate solutions of your ODE. Again, our quantity of interest would be the rise time, which is the time at which this thing is the steady state over 2. Okay. Now for a much more complicated system, it turns out that you cannot, in a closed form, solve this ODE. So we need to do some more work to do this, like to figure this out. And you can. And more or less what you can do is, you got the details in the lecture notes, but you see that the first phase will be very quick because you're not hitting a target that is as high as beta over gamma. And then the second phase will be slower. So just to make this a bit more explicit, phase 1 is kind of x being much smaller than kd, and phase 2 will be x being bigger than kd. And of course, there's a transition going on, but you can make everything precise here. And for much smaller than kd, what you will get is just our ordinary differential equation as we had it without any regulation, because k is so small as in relation to kd, so that this is more or less 0. And then you're rising up until beta over gamma, but to hit your target of xst over 2 for the regulated system, that's way too high. So this will have a rise time until it's finished of phase 1, which is, turns out, much smaller than 1 over gamma. And just a reminder, 1 over gamma was our basal rise rate in the non-autoregulated system. Then there's phase 2. So phase 1, you can work out that this is really quick. Of course, using these approximations here. So what about the second? In the second phase, we do another approximation. Now we're saying that the x over kd part is much higher than the 1 part in the denominator. So we're saying that our approximate ODE now is beta over x over kd. Okay. Just because if you divide here by kd, this means that this is much bigger than the 1 that is here. So you kind of forget about the 1. Again, you can make everything precise here by taking the correct big O's. OK, and this is the phase 2. So we're starting not at 0, but let's pretend we're starting at 0. You can solve this. Like, this can be explicitly solved. There's 1 over x. You can integrate that to the log. And you can integrate this to x squared. And what you do get is just for the second phase, just for this approximate system, is x of t will be the steady state times 1 minus e power minus 2 gamma t. Taking the square root here, but you're also having twice the rate in the exponent here. And this is important. So if you're setting this to being 1/2, what you do get is a combined rise time, only for the second phase, but again, the first phase doesn't really matter. It's much smaller, the rise time is much smaller, of I think it's 2 times log 4 over 3 over gamma. Again, this is approximate because there's a plus, a term here plus, and it's much smaller than 1 over gamma. And this you can figure out, this is like around 0.14 over gamma. And if you compare this with the rise time kind of the basal system, so unregulated system, this was log 2 over gamma, which is around 0.69 over gamma. Okay. So we're in the same order, but we are like 0.6 over 0.14, which is around 5 times quicker in the regulated system. Okay. You can see that. We did approximations here, but you can see this if you numerically integrate. You can see this in graphs. So this is the first graph of regulated system versus non-regulated system. Again, here, you don't, like, there's our analytical solution, which is this here, and a negative autoregulated solution, which is the numerical integration of our ODE, of our original ODE, and here's the unregulated system. Of course, here you see the unregulated goes much higher. This was to be expected. We set it up that way. We want to have high autoregulation. But now we're normalizing again. So this big steady state of the unregulated system, now we're bringing it down to 1. And here we see exactly this. We see that kind of in the normalized system, the regulated system is quite a bit quicker than the unregulated system. Which could explain why negative autoregulation is much more prevalent than you would expect in bacteria. It can turn up your rise time, just 5 times quicker. Seems like a good deal. Okay, before turning to positive autoregulation, do you guys have any questions? Anything that wasn't clear? Or if you want if you want more details on some of the things. That's good? Okay. Cool. This was negative autoregulation. There's two things. As does turning up gamma, which brings down your steady state and relatively makes your response quicker. Positive autoregulation is a bit different. What positive autoregulation allows you to do, and I might turn to the online notes here, because there will be a lot of graphs. What they allow you to do is to do something that's called multi-stability or bistability. They allow you to have your system either in one state or in another state, and stably so. More or less you can think of like you're positively upregulating yourself. So it means that if you have nothing at all, there's nothing there to upregulate you, so you will stay at 0. But if you have a lot of it, like because your autoregulation keeps you up, it will be very hard to drag you away from the high state. Because as soon as somebody drags you a bit down, you positively upregulate yourself, so you're pushing yourself again up towards the ceiling. And just a mathematical interlude here is something that is needed to properly appreciate bistability in genetic circuits. It's what's called Hill functions. Hill functions are generalizations of what we already seen. So maybe let's take the second one first, what's called the repressive Hill function. So here would be activation or production rate of a certain x, of something as a function of its regulator x. And here it's k power n divided by k power n plus x power n. We've seen this already. For n equal to 1, this is exactly our Michaelis-Menten regime that we had. Why? Because just n here, n equal to 1, so the n disappear. You can divide by k in the numerator and the denominator. So you get 1 over 1 plus x divided by k. So we've seen this. Just now, we added an n. OK. This is a repressive Hill function. And there's also an activating one, which is now we want a factor that goes to 1, that approaches 100% when we turn up x. This would be positive regulation. And this is just, you copy this, but now you're replacing the k here by an x. What does this do? If you're increasing the n, you're getting two things. Again, a trade-off. You would have your system, and so here we have multiple different n's here. So you have 1, which is this, 2, which is this, 10, which is the very steep one. If you're turning up the n just here by looking at the graph, is you see the system reacting later than it would have with a smaller n. But when it does completely. Like it's a very sharp transition from 0 to 1 here. If you let n go to infinity, you do recover just a step function. This can be explained, and I think we'll discuss this in an upcoming lecture. It can be explained from lower level principles by modeling this as a cooperativity. What that would mean is if you have, let's say, an activating Hill function of exponent n, and n is an integer, you can derive this equation by saying, oh, one copy of X doesn't suffice to activate your gene, like turning up your gene, but you need n of them. 1 does nothing, 2 does nothing, 3 does nothing, and then the fourth one does everything. Now you're turned on. What this means is you're reacting slower because now you need more of the X's to react. But when you react, you can work out the method, then you react very, very abruptly. Why do we need that? Because positive autoregulation enables bistability, but only if we're turning up the n. We'll see that in a bit. So positive autoregulation is we're having a gene, produces something, and this something upregulates yourself. The equation is not a mystery here. We're taking now the Hill function, so this generalized Michaelis-Menten dynamics with the power n. Of course, leaving as it is the degradation gamma. This is one set of parameters. Beta 10, gamma 1, k equal to 4, n equal to 4. And what we are plotting here are two different things. We have the degradation rate in orange. Just a linear function of x, gamma times x. And here we're plotting the Hill function. And we know that our possible steady states are, now we have multiple, is where the production rate equals the degradation rate. At these specific points, we have a derivative of 0. If we start there, we're staying there. Here it turns out we have three intersections of the two curves. And let's talk about stability. So I talked about bistability. And let's look at maybe this here. So here, this is a stable point. So if you start there, you stay there. If 0x, you stay at 0. And let's say now we're increasing a bit the x. So we're going a bit like 0 plus an epsilon. What happens here is that the degradation rate is still higher than the production rate. So we slide back here. On the other side is, again, a stable point. If we're going up a bit, we have higher degradation rate than production rate, so we slide right back. If we're going down a bit, now we have a higher production rate than degradation rate, so we're sliding up again towards this point. And this is what stability is all about. If you have like a small perturbation, you go there again. This in the middle is an unstable point. If you start exactly there, you will stay there, but if you're starting a bit below, now we have a higher degradation rate than production rate, so we slide here. And here, the same thing. If you start a bit above this unstable point, you're sliding up. So this is a bistable system. And these kinds of graphs where you plot the degradation rate against the production rate is a very handy picture to have plotted in front of you to assess bistability. And this here is the same picture, except now we turned n down to 1. It's not 4 anymore, it's 1. And what happens here is that actually you do lose bistability. So these production rates, they tend to look like this, or they do look like this. You have two intersections here only. This one, let's figure out what this is. If you're going a bit toward the right here, now you have a higher production rate than degradation rate, so we'll go here. And here, of course, you have a higher degradation rate than production rate, so we go back. So this is a stable point, but this is not a stable point. So you're not bistable. And of course, you can work out that this stability is, for most parameter ranges, they go stable, non-stable, stable, non-stable, and so on. Except if you're just tangentially hitting the line, the degradation line. And here, for example, we have two different parameters of gamma. Just to tell you, so now we're even up to n equal to 10, super sensitive. but it still depends on your other parameters whether you're bistable or not. So with this one, this is gamma being equal to 1, you will have 3 intersection points. Bistable as you before, this is stable, this is stable, this is not stable. Totally good, you're bistable. Here, now we're turning up gamma equal to 2, much steeper line here. Now we're hitting nothing except the origin. This is of course a stable point, but there's no bistability going on. You're always like going to 0. So why would you want a bistable system? What can that do for you? Bistability can build you a memory. It knows its history. Why? Because if you can push your system towards, let's say, the lower of the two stable points, it will stay there, even if there's some noise going on down there. If you're pulling it up, it will stay there, even if there's some noise going on up there. So you're building a memory, a 1-bit memory, also called just a toggle switch. You can switch it on, we stay on. Switch it off, we stay off. And this is one of the first breakthrough genetic circuits that was presented 25 years ago. And what they had is that they didn't have like positive autoregulation, but they have like mutual negative regulation of two genes. One gene X, one gene Y. You can call one of them on state and the other off state. And what they do is like if gene X is produced a lot, then we turn off gene Y. So gene Y will not be produced enough to repress gene X. And the other way around. So one of them mostly is dominant. You can, of course, work out the math. So here's just we're back at repression. So we only have the X's and Y's in the denominators. You can work out the math there. Maybe just one picture that I would like you to keep in mind is what's called the null clines. I think there is a very nice equation up there. Yes. The null clines for X and for Y, you have two of them, and there are two lines in the XY space. So X being one gene, Y being the other, and it's exactly where X will have a steady state. It's the null cline for X. And Y, the null cline for Y is exactly where Y would have a steady state, where its derivative will be 0. So this will be the degradation rate. It's normalized here. This will be the degradation rate for X, here for Y, normalized to 1. And this will be the production rate for X. And this will be the production rate for Y. And if this is 0, if this equation is fulfilled, then the derivative of X is 0. And the same thing for Y. And now you can plot these as you can play around with the parameters a bit. So here is the N, here are the betas, so the production rates. And what you need for bistability is intersection points of these things. So you need 2 of them. So all intersection, every steady state is an intersection point here. Here we have 3 for these specific parameters. With this one, which is stable, with this one, which is stable, and this one, which is unstable. And you can play around with the parameters also to show that it's not always possible to be bistable. Or at least it is possible to turn your parameters such that you're not bistable anymore. So here, for example, we're turning down the production rate of Y quite a bit. Then it always X would dominate. It would always go to this single stable steady state. Yeah, so this play around with also if we're turning N down to 1, we can come down to only one intersection of these. It's of course the 50-50 point. As soon as we're turning it up here, here around 1.8 something, we have these two stable steady states and the unstable in the middle. Right, and yeah, and it turns out that this is a very nice model, but it also really works. Yeah, sorry.
Is it binding stability? Do we always have a two stability point and one unstable? Or is it possible to have like just two stable points?
You can have them, but it's very rare in some sense. So let's put ourselves in this picture where here we have the degradation rate, and here we have the production rate. If you're doing something like this, it's always stable. Unstable, stable, unstable in this case. You can have two of them in a row, but it would be something. You would need to touch the line in exactly one point. But in most cases, you have stable, unstable, stable, unstable. Okay, yeah, actually I was already wrapping up here. Yeah, just to say that this model of this toggle switch can be made mathematically, of course, the analysis can be made. It works really well in practice. There have been improvements upon this toggle switch, that it works even better, but even 25 years ago, it worked pretty well. And kind of the math can give you an intuition of why that is, and even give you some parameter ranges of why your system maybe doesn't work. It's not bistable. It doesn't have an on and an off state. So what did we discuss today? I want to wrap up. We discussed genetic control.
We have one question.
Yes. Yes, please.
So this bistability means two steady states, the bistability, if it's always two steady states, and is degradation rate always linear?
Good question. So degradation rate, it can be very well approximated by linearity. Yes, because the most striking, the most important component of it is just dilution because of growing cells and dividing cells, which in many regimes can be assumed to go up linearly. The other bistability, yes, bistability means you have two different stable steady states. If you have three of them, it would be tristable, multi-stability in general, of course. But it refers to the stable states. Okay, thanks for the question. Yeah, just wrapping up. So what we discussed today? First, genetic control, what it is. You do have repression, you do have activation. Repressing yourself can speed up your response time. So if you have negative autoregulation, it can speed up your response time of your gene. If you have positive autoregulation, for some parameter ranges, you get bistability. You can build a toggle switch, a memory in your cell. Next time, we'll talk more about like Boolean logic, how you can use these things to build not only small components, but really build a Boolean circuit out of them. Okay, if there are no other questions, thank you. That's it for the lecture part then. Then we go, I guess, to the problem session. And yeah, online we meet again, I guess, in 2 hours-ish for the solutions. But of course, you guys, whenever you need us, you tell us.